All right. Good morning, junior woodchucks. This is a lab where we have extracted nematodes, and over there you can see the apparatus that we used. I'm going to go ahead and turn off the light. I've taken soil samples from 3 different locations in the UCA Nature Reserve and put them into a filter. It's basically just a coffee filter folded into a cone. And I put that cone in a funnel, filled the funnel with water, and soaked the soil, got it nice and saturated. And then for the past 28 hours, I've been shining a bright light onto the surface of the soil. What that'll do is the bright light, the heat, and the evaporation drives any nematodes that are in the soil downwards. And eventually they pass through the filter and pass into that rubber tube that you can see. And when I unscrew this little pinch clamp, like so, a little bit of water drains into this little beaker. So there's what the gizmo looks like. You can see there's the soil in a paper coffee filter. There are more sophisticated things I could use, but why? Everything's in a funnel, and then attached to the funnel, you've got this length of rubber tubing with a clamp on it. And so I saturated the soil with spring water, made sure that this entire tube was filled with water, as well as the funnel, and then put it up here for 28 hours under bright light. And I've done that with 3 samples, one of them from an oak-dominated region of the nature reserve, one of them from a prairie part of the nature reserve, and one from a more mixed forest portion of the nature reserve. And I do believe this one is the oak-dominated part of the nature reserve that we have here. So what I'm going to do is plug in the camera. And actually, before I'm going to do that, I just got a beep, but okay, I don't see that anybody's joined. So I'm going to plug in the microscope camera, and I'm just going to make a wet mount of this water here. And if everything has gone as it should, the water should have nematodes that have been driven out of the soil sample, driven down into the bottom of the funnel. So let me get a pipette. And you can't see it yet, but I'm just doing a regular old wet mount. I might end up doing a couple of these. But here we go. And give me a second to get the focus on this right. Okay, there we are. And I'm gonna, okay, there's one. Ooh, goody, there's another. Very typical here, nematode locomotion. You might remember we talked about this in lecture. that they don't have circular muscles, so they can't inch and they can't do that peristaltic motion the way that your typical worms do. All they've got is longitudinal muscles, which means that the only way they can move is by thrashing side to side. And so this is very typical of nematodes, this constant coiling of the body and bending it into C shapes and S shapes. This is pretty unmistakable nematode behavior. So let's see here. Yeah, there's another one right there. That one's got a little bit more inside her, so that may be a female. In some species, the females have blunter tails. That's the one we just saw. So I've got two here. Oh, good. Mercifully, that one's moving a little bit more slowly. All right. Good morning. We are looking at nematodes extracted from soil at the UCA Nature Reserve. If you missed how we did it, you can you can have a look later. This is being recorded. And this one's moving very slowly, so I'm going to zoom in on somewhat higher power. And let's get a close-up look at this bad boy, or girl, or possibly both, depending on species. So that's the tail end. And over here, if I can move this in the right direction, you can see the gut running all the way through. You could trace that back to the anus, which is not at the tip of the tail, but a little behind it. Oh, and this is important. It can be tricky to get them to slow down long enough to spot this, but if I get the focus just right there, if you look in the head, there, I'll try to center it. Come on, you silly nematode. There, if you look very carefully in the head, right back of the tip, if I get the focus just right about there, there is something inside the head that looks like a nail. It's showing up darker in color the way that we've got the lighting arranged. And it's pointy at the very tip and goes back a little bit. And at the back end of it, there's a little broad area that looks a little bit like a nail head. So let's try that again. So yeah, right there, if you look in the head. There it is. What that tells you is that this nematode is a plant parasite. Most of the nematodes that we'd find in soil are free living. Some of them can prey on fairly large other invertebrates like other nematodes. Many of them have just kind of a tubular mouth and just slurp up bacteria. But that nail in the head right there, we've hit the nail in the head, as it were. What that tells you is that this is a plant parasite in a group called, I believe it's the Telenkida. And what these do is use that as a piercing stylet to pierce plant root cells and slurp the contents out. So what I'm going to want to do is record that that is a plant parasitic nematode. Actually, if you'll give me just a sec, there's something I need to look up here. Okay, here we go. I'm going to, let me see, I'm going to show you something here. I'm going to present a different window. And there it is. This is from the handout that I'm going to give you guys. There's 4 major families of soil nematodes. Over on the left, that's what a microbe-feeding nematode looks like in the family Rhabditidae. You can see that the second one, predator on invertebrates, has those teeth inside. And then that third one, Tylenkida, that dark object right there, just inside the head, that's the nail type thing that I was telling you about. So what we want to do is count the nematodes and identify as many as we can as either rhabditids with the tooth mouth, diplogastrids with that pair of teeth, tylenkids with a nail, or dorylamids, which tend to have a much longer stylet that they use for parasitizing invertebrates. So we can get an idea of how many nematodes we have of each type. So getting back to where we were, did everybody see that? Did everybody see that diagram I tried to show you guys? Okay, Jamika says she did. Great. This morning, once we're done here, I'm going to send you copies of that directly by email, and I'm sorry I did not do that beforehand. But you'll be able to refer to that when you're making your judgments. Okay. Good morning. Sorry you dropped out there. All right. So let's go and look at more of these suckers and see what we can find. So we've got this one kind of sleepy one right here. This is the one we were just now looking at, and we determined that it was a plant parasite. So I'm going to go over here to the corner of the slide and just go back and forth and see if we can spot more. There's at least 3 on the slide and probably more than that. And if I need to, we can make more wet mounts, of course. Okay, there's one. So let's zoom in and see if we can tell what sort of beast we are dealing with here. Of course, the tricky part of this lab is that since they do thrash a bit, it's not always straightforward to tell what type of nematode you've got. All right, I'm going to see what I can see looking through the eyepiece. And I may be smoking toads. Sorry, old expression. But I think I see a nail in that one. So I'm going to call that one another tylenkid. So I think that's a second plant parasitic nematode. Oh, that's a little ciliate. We do have them in soil. And we're panning, panning, panning. Do, 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 do. Not sure what that is. There's that one that was just thrashing. Oh, there's a second one right there. That's not one that I think we counted. And this is actually outside the slide, so if they're thrashing around out from under the cover slip is what I mean. This is outside the cover slip. And if they're there without the cover slip holding them flat, it can be really hard to get them in focus. So I think by the power vested in me by the state of Arkansas, I'm going to call that one undetermined. So keep track with me. We've got 2 Tylenkidae and 1 unknown. And we pan and we pan, diddly diddly. There's a little ciliate for you again. There we go. Here's a 4th. And fortunately, it's moving slowly enough that I'm actually going to try invoking the highest power. And let's see what we can see. Okay, there it is. Try to get it centered on the head. Okay, that's another parasitic nematode because right there, as I focus up and down through the head, you can see that stylet that Telinkid nematodes have. Right there, right inside the head, you've got that long nail. Right there, at least I can see it, and I haven't had any flashbacks for 3 whole days, so I'm assuming that it really is there. Sorry we lost him. Let me try to find him again. Or her, for that matter. Or they, for that matter. There. I can't get him in the center, but, or her. Okay, there's a good shot of the head, and right there, yeah, you can see that nail. Okay, so that is a 3rd potentially plant parasitic nematode in the family Telenkidae. And thank all the gods it's moving slowly enough that I can spot that. But yeah, there it is. And there's that nail right there. Okay, so we've got 3 that do have that nail, and 1 that we couldn't be sure. All right, that's the one we were just looking at. I'm out of focus. Hang on. Yeah, there's the one we were just looking at. Okay, here's a new one. And let's see if we can get a brief look at its head. And since it's kind of moving in a helix shape, it's I can't get a good steady focus on the head because it's kind of moving in and out of the plane. Okay, but I can convince myself if I squint. If I look very closely at exactly the right moment, yeah, okay, I can convince myself that I'm seeing that nail right there, right there, right there, right there. Yeah, so that's going to be yet another plant parasitic taxon, I'm pretty sure. So what are we? 4 plant parasites and 1 undetermined. All right, there is yet another. Okay, hang on. I've got something to mess with on my computer. Okay, good. That went away. Okay, so yeah, that's yet another. Let's zoom in on that one and see what we can see in the head. And this one, again, it's hard for me to get a good tight focus. Ah, little ciliate went by. Good to know they're there. Yeah, because this one is sticking its head almost directly away from the direction I'm looking in. So this one is just being annoying. Um, I thought at one time that I could see a stylet in there. Okay, maybe I can. Yeah, my judgment call is going to be that that is a tylincid. So I think we're up to 5 plant parasites. Although really, let's make it 4 and 2. I'm just not sure enough about that one to really feel comfortable with it. So let's just call that 4 tylincids and 2 unknowns so far. And oh, good. Here we've got 3 that have mostly stopped moving. So maybe we can knock these out quickly. That one does. Yeah, you can see that nail right there that shows it's a telenkid. Boom. Yeah, even back of the nail, you can see the little esophagus kind of making a little S curve. So what are we up to? 5 and 2. And then, I'm going to have to look this up. I can't remember what this is called, but this is a microbe feeder. It's not a plant parasite. And there are some nematodes that have weird little cuticular outgrowths on the head. So this one, should actually look like it's wearing a crown or something like that. And I'm suddenly drawing a blank on what the genus is, but I've seen these before in the nature reserve. But yeah, I'm pretty sure that's not a plant parasite. That's a microbe feeder. And if we could see this in 3D, it would look really cool. The thing has little, kind of 3 triangle-shaped wings on its head. So we've got one of those. And what are we up to? 5 microbe feeders. And then this is the 3rd one that I spotted. And if that one's a juvenile, it just may not be possible to tell. I guess maybe there's a little stylet, but I'm going to put that one again as uncertain. So what are we up to? 1 microbe feeder, 5 plant parasites, and 2 unknown. Okay, those are the ones I was looking at. All right. Okay, here's one. Okay, that's there's the nail. All right, so that's a plant parasite. So we're up to what? 1, 6, and then 2 unknown. All right, there's another one. Okay, I'm barely seeing a little nail right there. But I've been calling those unknown. I think most of our unknowns might just be juvenile plant parasites that don't have the mouth parts developed. But just to be consistent, let's call this, what are we up to? A 3rd unknown? I think this is what we've got, 1, 6, and 3. All right, here we've got yet another. Okay, there's the nail. So I'm going to call it we're at 1, 7, and 3. And there was one down here a minute ago. Yeah, there's the nail. So 1, 8, and 3. All right, we've got rather more plant parasitic ones than I was expecting, but so be it. And And 3 of the suckers have just hove into view. So there, yeah, there's that nail. So what are we up to? 1, 9, and 3, I think. You know, if I was smart, I'd have a piece of paper and be keeping tallies. Hopefully you guys are doing that. And one over there. Okay, there's a very faint suggestion of a nail. Let's call that 1, 9, and 4. Yeah, same thing here. I think the juveniles are probably telenchids, but I can't be sure. So I'm going to say we found 1 microbe feeder, 9 telenchids, and 5 uncertain. And remember, folks, if you decide you'd like to do this for a living, the state nematology office in Hope might be taking resumes. Worth a shot. Okay, there's one that I think is pretty much dead. But let's see what we can spot here on this one. Okay, that's a second one of the microbe feeders that has those funny little wings on its head. So let's say we're at, what are we at? 2, 9, and 2, 9, and 5, I think. But yeah, that one is not feeling very good about life at the moment. Oh, there's that's Colpoda. Actually, a pretty common ciliate in soils. That bean shape is very distinctive. Some of you saw it in Lab 2, Colpoda. But there was another nematode right here that is slowed down a bit. Oh, that's a male. The end that's down at the lower part of the field of view, that tail looks a little bit different. That's actually the copulatory bursa, and I can see the spicules inside. So that's a boy nematode, and I'm not seeing any trace of that nail-looking thing, so I think this is another microbe feeder. So I think we're at, what are we, 3 microbe feeders, 9 plant parasites, and 5 that we don't know. So, yeah, that's cool. That's a male nematode right there. And we keep looking, and okay.
I'm sorry I had to leave. Where are we with the count?
Hopefully I haven't lost too much count, but I think we're at 3, 9, and 5. 3 microbe feeders, 9 plant parasites, and 4 that are, I think, are juveniles that I couldn't be absolutely sure. If I'm wrong, somebody can put it in the chat or something. Oh, this is odd. Okay, that's another. That is prune. What the heck has that thing got on its head? Okay, I'm not seeing an. I don't think I'm seeing a nail. Yeah, I think this might be another microbe feeder with a. Well, maybe I am. It could just be. I don't know. I think this may be a species that just has a different set of deedly bobs on its head. They can vary like that. Here, get you in the spotlight there. And it's active enough that I can't really get a fix. Okay, by the power vested in me by the state of Arkansas and having a PhD, I'm going to call that a microbe feeder with deedly bobs on its head, but different deedly bobs from the ones that we've already seen. So I'm going to say that we are, what, at 4, 9, and 5. I could be wrong about that. Not that I've ever been wrong before, but I guess there is always a first time. That one I think I've looked at. This is one that I haven't. No, I think I have. This is the one that wasn't moving. Okay, and that's going to be a juvenile, but I don't think I'll be able to tell. So let's call it, what are we up to? 4, 9, and 6? And there's one that's trying to sneak out from under the cover slip. Sneaky little nematode. And that's got a nail. So 4, 10, and 6. There's the head is just crossed under. And yeah, I can see the nail right there. Yeah, that's another plant parasite. So we're at the count of 4, 10, and 6 is how I make it. I think we're almost done. Yay! All right, real quick, I think that's going to be another juvenile. Yeah, I'm going to call that one 4. Let's call this 4, 10, and 7. Because I can't convince myself of what I'm seeing. And there's a grown-up nematode that's trying to escape. Okay, that one. Yeah, that one I just can't see. Yeah, and that's another real small juvenile. Although with that one, I can't see any trace of mouth parts in there, so I might go so far as to say that's reasonably likely to be a microbe feeder. So I'm going to say that's, where are we? 5, 10, and perhaps somebody who's writing it down type that in the chat. Here's a big one. There's the nail. So that's 11 plant parasites. And I think we're at 5 microbe feeders, 11 plant parasites, and however many that I couldn't tell. But yeah, there's that nail. That's obvious. There we go. Good little nematode. And that would be that. So yeah, I think our final count is, what have we got? 5 microbe feeders and that last one was a plant parasite. So we're up to 12 plant parasites. And then help me out. We're up 5. Okay, let this be a lesson to you, is to make sure that you're writing this down, because I didn't have a tally sheet. So I might have to go back and watch this again. My data is going to be suspect. And 6 unknown, Shivam says. Okay, so we ended up with what? 5, 12, and 6. All right, what I'm going to do is I'm going to stop recording. You've seen what needs to be done. And I'm going to go ahead and record samples for the other 2, although you don't necessarily have to watch these live unless you really want to. And I'm going to have you guys do the count on the video and try to work out the proportions of microbe feeders to plant parasites in these 4 families that I'm going to send you guys in a handout in just a couple of minutes, in fact. So what you're going to do is watch the videos, try to count the number of different nematode families. And the cool thing is that I've actually got some data going back quite some time, for several years. And I want to see if we can record any kind of long-term change in nematode assemblages, or if they're staying pretty much the same. I don't really know what we'll find, but I've got the data. We might as well subject it to some kind of analysis. Okay, everybody figured out what you're going to do? I will take that as a resounding yes, and I'm going to go ahead and stop recording.